Okay, fantastic. All right, well, thank you so much, everyone, for joining Jane and I tonight. I'm doing the speaking honors, but Jane will probably pop her head in now and then to add some colorful commentary, which will be good fun. So I have a caffeinated beverage. I hope you guys are enjoying something on our date night tonight. It's lovely to have an evening with the wildlife heroes out there because we really respect and honor everything that you do. So we'll just get on with things. So just to overview what we're going to be talking about tonight, we thought we'd just introduce you to a little bit about our program. Some of you might have heard of us, some of you might be very familiar with us, but we'll just start off with that little introduction. And then we'll just talk a little bit about what we do and how we do it. So the processes, it's not all magic behind the scenes. And we just hope to share a little bit about that with you. It might explain some of the things about why we ask strange questions and why we're very fussy about some of the things that we can help with and some of the things that we just can't help with. It's beyond us. And then we thought we'd share a few recent disease investigations and where we are in those. We're not finished all of them, but we're certainly down the track on quite a few. And it's just an honor to be able to present that back to you and get some of the science back to where it can be really used on a day-to-day basis. So the registry, we're a wildlife conservation research program of Taronga Conservation Society Australia. We provide a diagnostic service for free-ranging wildlife, and then after doing that for close to 40 years now, we've got a wealth of information and resources on wildlife health. Really helps us to put into context what's new, what we've seen before, and how things are changing over time, which is a brilliant little window into ecosystem health. So tonight, I guess we'll start off with this. We work with a lot of carers who want to see what we're seeing and want to know more. So we've included some photos tonight that some might consider to be a little bit graphic. Again, everybody is different. We appreciate that not everybody wants to see this. But we do want to share what we're seeing. And if you might be a little bit squeamish about some of this stuff, you might want to just listen along and then save the video or turn off your video. But we do this with the most respect for wildlife because we want to make people aware of exactly what we're seeing. We've tried to make sure there's absolutely no gratuitous imagery in there whatsoever, but just so you know what might be coming. So our role, we have a mobile veterinary lab that we take all over the place and a little veterinary clinic that we can go along as well. So over here on the right, you can see our portable anesthetic machine. We can set up a table on Christmas Island, on Norfolk Island. We've been around the traps, Cocos, Booderee National Park. We have a service level agreement with Parks Australia, so we help them with any health concerns they have on any of the federally managed parks and with animal welfare. So Kakadu, Uluru, Christmas, Cocos, Norfolk, Booderee National Parks. So we have to be able to respond. On the bottom left is our little whale necropsy kit as well. So we just sort of stand ready, ready for action, no matter what might come at us any day. And you never know what's going to come on any given day. So the program, on the front cover, I had Dr. Hannah Bender's name because she's been integrally involved in all of the investigations that we're going to talk about tonight. Sadly, but great for her, Hannah's found a very attractive position elsewhere, and she's moved on from our role. So I don't have her photo here, but we love Hannah, and we're thinking of her, and she knows that because I just texted her. So we're very fortunate. It's just Jane and I really at the moment, but we're very firmly supported by the love and care of a number of volunteers. So I see Ann Jackson on there. She's awesome around Taronga, and we really value her assistance. And we've also got some fantastic volunteers. Peter Johnson is a retired vet with the New South Wales Department of Primary Industries. He was in today helping us out. We've got Bryn Larndar, who's a veterinarian who had a clinic up in the Pittwater area, who comes in quite regularly and just is incredibly helpful. And then Karina, she's in the middle here. She works at the Brain and Mind Institute in their histopathology lab, and she comes in every other Friday to help us out. Kath over here, Antarctic, she runs our communications programs, so just single-handedly, which is absolutely phenomenal. So this is Jane down here, and then we've got 3 of our PhD students. So we're just finishing off on a whole swath of PhD students. We've had about 6 for the last 3 years, but they're all just in the process of finishing off. But they're the ones who've done a lot of the work that we're going to display for you tonight. We've been really blessed with brilliant young scientists to help us out. And yeah, we've been really, really fortunate. And the other people that we're very fortunate to work with are Paul Thompson and Natalie Miller down in Taronga Wildlife Hospital's laboratory. So I'm going to show you up close and personal in a little while some of the things that they do, but they're responsible for doing the zoo collection animals and also some of the wildlife that we bring through. So they're mostly looking at blood samples, at poo samples, looking at parasites, and they've got a farm for bacteria and fungi, and they're just brilliant. They're the kind of guys you can go down to and say, oh, what have you seen? I've been seeing this. And yeah, we get right into it with parasites, and especially parasites and fungi. So these are fungi. Some of the work that we do is for and with the Environment Protection Authority. You might have heard about, oh, actually, I'll talk about that a bit later. So when there are mass bird mortalities, we're never really sure whether it's potentially an infectious disease or whether it may be an accidental or malicious poisoning. So we tend to have a look at the animals. We'll send samples back to the Environment Protection Authority for testing. Other samples will go down to the Agriculture Department to rule out avian influenza, West Nile virus, Newcastle's disease. And then if it's none of those, well, then we start our diagnostic process to find out what might be going on there. And we've actually, with the help of another PhD student, identified about 9 different viruses in some of these birds that are submitted to us as likely intoxication. And we found out they weren't. Some of the research that we get involved with is spin-off. If we find something new and we need to figure out what that is and what the risks are for threatened species, for wildlife health in particular, and for human health, agricultural animals, and then what are the risks of that to the environment, and how can we potentially mitigate some of those risks. Some of the research projects are little and some are big, like is disease a factor driving multi-species extinctions on Christmas Island? That was a big one. Took us a little while to get our head around how to frame that up, but it's been really rewarding, and we're still working on Christmas Island from 2010 to now. Education, we try to, because it's such a wealth of resources that we're so fortunate to be surrounded by. We get to see so much fascinating stuff. We really try to get that out to wildlife carers and veterinarians as much as we possibly can. And it's hard to do it on a piecemeal basis. We really need to structure that in a framework. So in 2008, 2012, 2019, I'm not sure, it's probably every few years, whenever we get up the energy to do it again, we host these big courses, and we try to bring in about 6 overseas veterinary pathology specialists. We go through each taxonomic group of animals and all of their diseases. So it's pretty mind-blowing, but it's also just a fascinating thing. We tend to get about 150 participants, and many of those are from overseas. We tend to run scholarships for veterinarians from developing countries to join us. So it's just a really lovely environment to learn. So we'll have to do a post-COVID one coming up. Because it's just 2 of us and we've got such like bombarded by stuff coming in, we really have to have a system for information management that tracks everything, so that we can go back when we get requests. Someone's doing a textbook chapter, or they see an emerging disease, they want to know all of the diseases of our particular species or a particular group of species, or they want images of this, that, or the other. So we've got a really fantastic online information management system that tracks all of our digital images. Even we've got this fantastic slide scanner that scans glass microscope slides at multiple planes, so you can interrogate those online through the system. So it's kind of cool. We can map things out. We get involved with outbreak investigation, and it used to be that we'd do one every couple of years. We'd have something cool, and it was really in a common species, and unfortunately it's really escalated over the last few years. So we're having something new many times a year, and often we've got large outbreaks that are happening concurrently, and more we're dealing with critically endangered species. Either they were critically endangered before the outbreak started, or through the course of the investigation, they became critically endangered. So this is a healthy Bellinger river turtle. And over the course of a 5 to 6 week period, they were pretty much driven to functional extinction through an emergent virus. We've talked about that a whole bunch of times, so I don't want to bore those who have heard the story. So I've given you a lovely, healthy Bellinger river turtle here. But I guess the tricky part for us, and probably the part we enjoy so much, is because when something's really new, there are no tests for it. You have to go back to this combination of first principles and cutting edge science in order to try to figure out what's going on. It is the world's biggest puzzle. And when you have an animal like this Bellinger river turtle that you don't know that any vet has ever had their hands on before, no baseline data at all, it's really challenging. And yeah, that's probably why we love the tricky jobs. So we've started to try to communicate with individuals such as yourselves, the wildlife heroes out there, a little bit more regularly. So we have started up a Facebook group, and this is the part that Kath is managing, just trying to communicate things like this. You know, it's been really wet over this autumn, or the lead up to autumn, so we've seen a lot of avian pox. Webinar series that are coming up, just things that wildlife rehabilitators and veterinarians who work with wildlife might be interested to know. Some of them are good news stories about wildlife rehab, but more, whenever we can, we try to make it about interesting cases and resources that we think are interesting that we hope that you might be interested in as well. And thanks to Kath for all her work with this, because it is brilliant. Well, we think it's brilliant. So one of the areas that we've been really interested to get involved with has been marine plastics and looking at some of their impacts. So this is a Risso's dolphin, and it was found on the beach at Curl Curl. And I think Mike Baird, the premier at the time, was surfing, and I don't know, I think there was a photo of him in the local newspaper with the dead dolphin. So there's quite a bit of media interest. And this animal sort of was a bit of a twofer as far as dolphins impacted by humans go. So I hope you can kind of see the bag, the plastic bag that we're pulling out of its stomach. The other thing it also had was toxoplasmosis, so a parasite that's transmitted through cat feces. And it actually survives pretty well in the marine environment. So don't put your kitty litter in the toilet, and tell your neighbors not to do that either. But this animal, it was just one animal. We struggled about whether or not to release this image publicly because it's pretty graphic, right? So in the end, we handed it to National Parks and Wildlife Service and said, well, we're doing this on your behalf with your animals, and they put it out on their Flickr account. And it was just at a time when there were a lot of community groups trying to solve the problem with single-use plastic bags. So it was brilliant. There were effigies on Bondi Beach, and a petition was started. They got over 15,000 signatures on the petition, and that led to the Senate inquiry for the use of plastic bags. So just trying to get some of this information to the community and some of the groups that could really use it has just had some incredible outcomes, which, you know, we're only a minor part of some of those outcomes, but we're still proud nonetheless. And sadly, we see some of the marine fauna that are affected by anthropogenic debris, recreational and marine fishing nets. And I think there's been some stories on the news about ghost nets and their impacts recently. So we see some of that, nothing like they see up in the Gulf of Carpentaria and further north. So it's still sad to see, but it's really good to document boat strikes, anthropogenic impacts. Jane has gone through recently and summarized all of our penguin mortality, and it's really amazing how much we can attribute to boat strikes, propeller injuries, just various human impacts, dogs. I'll get to that now as well. So these are some of the little penguins from the Manly colony. And this was a case quite a while ago. I called in on a Saturday because there was concern that these penguins had been shot. There were 8 on the beach altogether. They were just, just concerned there was someone wandering around Manly with a gun. And then there was also, later in the week, there was also a long-nosed bandicoot. What we found when we opened up the animals is that they had bite wounds inside. And what we tend to do is measure the distance between the individual puncture wounds, and then we can determine whether it's likely to be a cat, fox, small dog. In this case, they were really big distance, and it could have only been a large dog. So we got a little bit fortunate again to get Bob Debus, who was the environment minister at the time, interested, and that led to the declaration of critical habitat for the little penguin, which again is just a really proud outcome. It's not us, it was all the penguin rangers at Manly that just took hold of this one little event and just ran with it. So just really great how we can work together. I think we are working together. So now I've been asked to cover a little bit on zoonotic disease. And so those are diseases that transmit from animals to people. And this is a slide from my epidemiology professor at vet school. And he was a really dynamic fellow. He used to come in with rock music blaring from a ghetto blaster, and he'd have photos of the 4 horsemen of the apocalypse talking about disease transmission. But I guess this is another thing that struck with me, is that these are the things that you need to have inscribed on your postmortem blades, because these are the things you won't come back from. You know, other things are treatable and these just aren't. So pay really close attention to these 2 things. Inscribe them, have them engraved on any knife that you might ever use on an animal. So with anthrax, there is an anthrax belt. I'm hoping you can see that on this map here. So we really don't know what the impacts of anthrax might be on macropods. They're a ruminant, basically. They're a grazer. So we don't know. Just pay close attention if you have macropod mass mortality events in particular in that belt. With humans, humans can be exposed by ingestion, by air. Most often it's little cuts and scratches on your skin as you're handling an animal that's been affected, but you can also inhale the little bacterial spores. It's called the bacterium that leaves spores. And the problem with these spores is they can live in the environment for years. So once you've had an affected animal in a location, you tend to see it again the next year under the right conditions, like low oxygen and moisture. But if you happen to inhale these spores, you can get a pneumonic form or a lung form, and it's really nasty. So you really don't want this. It's a very fast replicating bacterium, and it enters the bloodstream incredibly quickly. Australian bat lyssavirus, I think you probably have heard an awful lot about this, so I won't go on at infinitum. It's just any bat in Australia, any bat anywhere in the world could have lyssavirus. And pretty much as far as we know, that all of this group of lyssaviruses are capable of causing the clinical syndrome of rabies when they get into a non-target species like a human or another animal. We can't tell which bats are affected. Bats have this capacity to harbor viruses for a long period of time with no outward signs, and then suddenly they can break with it. So we can test them all we want, a live animal, and never come up with a positive, and suddenly an animal can break with rabies just unexpectedly. So just please take care when handling bats. Use all the PPE every time. So psittacosis is a bacterial infection in humans that's caused by Chlamydia psittaci, which is this sort of deranged bacteria. It's kind of, I don't know, they call it an unusual bacterial form. In parrots and other birds, it's often called parrot fever, ornithosis. It's not just parrots, though. It's a real misnomer to call it psittacosis, because you can get it from anything: chickens, pigeons, absolutely any bird. And it's one of these things so highly infectious that one little organism can cause massive infection really quickly. So you just have to be super careful when you're handling sick birds. And take precautions. And it's one of those things, I don't know, I've worked in an avian practice before, went in through the zoo vet side of things, and you just had to be aware that psittacosis and ornithosis in animals, it can look like anything, because it can present as respiratory or gastrointestinal or neurological. It's one of those things that can do anything. But it can also leave lasting scars. So if you don't pick it up in the acute phase, it can become a really nasty chronic disease. And I guess at the clinic that I was working at, the receptionist, who was the owner's wife, she was really sick, and she was in hospital for 2 weeks before anybody clued in that she could have psittacosis. And she's the wife of the owner of an avian veterinary practice. So it's one of those things that can bypass us all. We don't really think about it. And if you don't tell your doctor that you think this could be happening, they won't test for it, and the drugs that they give you won't likely work against it. It requires a really specific antibiotic. So it's really important. I guess the vet receptionist, she did recover, but she is diabetic because of the damage to her pancreas was just lasting. So definitely worth keeping in mind if you get sick and you work with birds. Just talk with your doctor, make sure they roll that out. The other thing that's actually very similar, we call these little blue dot diseases because they're little tiny bacteria that live inside cells. I don't know, on this lower picture at the bottom, can you see all those cells are just kind of filled with tiny, tiny little blue dots? And that's blue dot disease. You have to figure out what that is. But at least you can see them. Viruses you can't see, and that makes it trickier. But tularemia is something that we were fortunate with our colleagues to identify back in 2017. It was the first time it had been confirmed in the Southern Hemisphere. It's a pretty potent zoonotic bacterium. There have been several cases, suspected cases in humans in Australia with people getting really sick down in Tasmania, a couple of people getting sick in Tasmania when they were bitten or scratched by ringtail possums. Another person in Tassie was kayaking and he was in water. So it's a bacterium that was thought not to exist anywhere in the Southern Hemisphere, but surprise, it's definitely here. We know it's in New South Wales and we know it's in Tassie, and we don't really know about where else it could be in Australia. We identified it in 2 ringtail possums, both of them from the northern part of Sydney, the same suburb, about a year apart. Both, we had only got 1 animal, but they represented mass mortality events. So just be really careful with this one. It's just like the chlamydia. It can do anything. It can present as skin lesions or respiratory infection or gastrointestinal, rarely, but it can be neurological as well. It's transmitted by ticks and in water. So just be really careful when you're dealing with sick animals. And again, if you handle sick ringtail possums and potentially other small mammals and you're not feeling well, then it's something to bring up in the discussion with your general practitioner. So now we're going to switch over and follow the yellow brick road, because this is the fun stuff. We're finished with the serious stuff. Well, this is still serious, but it's interesting anyway. So this is our pathway to achieve a definitive diagnosis, to know exactly what happened and why. Actually, that epidemiologist, he used to say that why is a religious question. We can only know the who, what, where, and how. So we'll look at that. So what we really need to make a good diagnosis is a good history. So, you know, all the records that you're keeping, that's awesome. It really helps us out to know how long animals were in care, what they presented with, where they came from, what species are affected. Good photos are incredibly helpful. They help us along the pathway as well. When animals present to us alive, we can take blood samples, we can count all the red cells, all the white cells, look at the differential cell count to see if there's toxic change in any of the white cells, which makes us think it's more likely to be infectious. We can sometimes even see those little blue dots in circulating cells to know that we're dealing with a systemic bacterial infection. And then we get on to the even more fun stuff, which is histopathology. So when we're doing necropsies, we take, I've got props today, isn't that awesome? So this is little bits from a little neonatal dolphin, which was sadly smashed probably by another dolphin. So when we're doing a postmortem examination, we put tissues into formalin. We make sure they're quite thin, no more than 1 centimeter thick, so that the formalin penetrates. And once those are fixed, then we put them into little cassettes and they go off to the University of Sydney for us. Thank you, University of Sydney, and everything you do for us. And they embed those tissues into wax. And what they can then do is take a little knife and cut off a little sliver and put it onto a glass microscope and stain it up. I don't know if you can see that. Can you see that? It's like a heart. I should have tried this before. So that was just individual little tissues. We had some bats with skin lesions a couple of weeks ago. So bats, its skin all along their patagium where their wing webs was horrific, and it was a lot of skin. So we rolled it around a little stick so that we could get actually quite huge amounts of skin on one little sample, because we don't have a lot of money, so we have to save money. And sometimes our blocks can get really big. So this is an eye, probably from a seal. And then we can get a really big corresponding slide to go with that. I need something white. Can you see the eye? Woo. So that's what it looks like. And then under the microscope, you get a much better look than that, obviously. But then we can look at the tissues and look at the architecture. Sometimes we can see bacteria, fungi, parasites in there. Sometimes we just see the kind of inflammatory change. If it's eosinophils, then it's likely parasitic. If it's mononuclear cells, lymphocytes, and plasma cells, then we're thinking a small intracellular parasite or a virus. And if it's neutrophilic, we're kind of thinking likely to be bacterial. So that's super helpful. And that lets us, we don't have all the money in the world to do every test on every sample that we get. So from the histopathology, that guides us on the next step. Do we do culture? Do we do bacterial culture, fungal culture, viral culture? And if we do those, here's some. Paul and Nat were awesome today. They gave me some props. So this is a fungal culture on a fungal plate. So we can grow the fungus and then try to learn a little bit more about how it grows and what it needs to grow. And then the other cool thing that they can do, which I think is a brilliant thing, is they can, this is a bacterial culture plate, but each of those little discs is impregnated with an antibiotic. So you can see there, can you see the clear spots? There's no bacteria there because that bacterium is sensitive to that antibiotic. So that's one you could use on an animal and it would work instead of having resistance. The ones where the bacteria grow right up to the disc, don't bother with those antibiotics. That's not going to work. So that's really cool. That's the kind of thing we can learn in about 24, get a bit of an idea of what organisms are there. Once you know the name of the organism, you kind of know what its nearest neighbors do. If it's E. coli, it's fecal oral. If it's Aeromonas, it's likely in water. You can kind of find out how to break the cycle just on the behavior of that group of organisms. But sadly, only 3 to 10% of all organisms grow in culture. There's this huge world of microbes out there that we have not been able to detect easily. So PCR, polymerase chain reaction, we can go and do specific tests for either specific viruses, bacteria, parasites, or you can do like a family level. You can look for all the herpes viruses, all the adenoviruses. And that's brilliant if you kind of know what you're looking for. But in these events where we don't really know what we're looking for, we don't have all the money to do all of the different viruses that are known to man. So I guess the thing that's been really revolutionary is going to metatranscriptomics, which I'll talk to you about in a second. But toxicology is also a really important factor, and it may be just that the animals are getting sublethal concentrations of toxins and it might make them more susceptible to other things, or we might have fatalities. We'll talk about some of those events with some of the cases we're going to look at. So metatranscriptomics, that's a brilliant thing that we've only in the last 3 or well, I guess maybe 5 years had access to. And that's working in collaboration with Professor Eddie Holmes at the University of Sydney. So he's one of the co-discoverers of COVID, and he was the one who pushed the button to publish the sequence data so that globally everybody could start manufacturing their vaccines based on that sequence data. So he already had a research project with the hospital in Wuhan before the virus hit. And just really prepared. So the process really, unfortunately, I don't have a really cool photo because it's a bit of a cryptic test and process. But they take a sample, they sequence all of the RNA in the sample, and can put all of the pieces, the fragments that they sequence back together using the big supercomputers at the university, and detect all of the bacteria, all of the fungi, all of the parasites. Pretty much everything, all the viruses. They're really keen on viruses, so you kind of have to work to get them interested in other things. But it's just a brilliant process. It won't necessarily pick up absolutely everything, but it's doing pretty well. Things that are latent, like that rabies virus, if it's not reproducing, it won't generate RNA, so you wouldn't necessarily pick up some of those latent things because they're lurking somewhere in the shadows of the animal's body. But these fragments of RNA, it's the same kind of stuff that the Pfizer vaccine is made of. So You know how sensitive that is when you're transporting Pfizer vaccine. It has to be at -80, and there's all these bells and whistles to make sure it's really fragile. It breaks down really quickly. It's not a molecule it's a transient molecule. It's not meant to be permanent in most circumstances. So just have to get supremely fresh specimens, and then we take the samples and put them straight into our -80 freezers. We transport them on dry ice, which is -100 degrees. We just have to be really careful. So if you find that we're just being really annoying by asking for really, really fresh animals, it's because we're really concerned about what might be happening. We think it might be something new, and we want to use this process for that. Oh, and on the other side, we have Meng Shi and John Sebastian Eden. And if you think we're nerds, I asked them for a good photo of themselves, and they gave me this. That's their photo. So back into some of the current things that we've been seeing lately, I suppose. So you all know there's a big mouse plague happening, and I would not want to be in that area. You know, I've been hearing about people putting buckets of water under their bed legs. So I don't know if some of you might be in those kinds of zones and just understand how.
Oh, Carrie, you just muted. Sorry.
Okay.
Perfect.
Yeah.
You're back.
Okay, so what we've been seeing is there's been some misuse, so unapproved use of.
Oh, Carrie, just happened again. Sorry, I'm not sure what's going on. I just muted you again.
Okay, it's not me.
Yeah, no, I can see. I'm not sure if anyone else is doing that, but yeah, you're good to go.
All right. So suppose that some of the mass mortality events are up to 100 birds in just one particular location. So I guess that one of the intoxicants that we're a little bit concerned about is zinc phosphide. And when it's applied normally, I think it's actually a really good rodenticide, and the risk to wildlife is probably the lowest of any of the chemicals. It's got approval to scatter it around in paddocks and in very small, granular form. It's something that most wildlife won't actually pick up and consume, and it tends to degenerate in the wild quite after a week or two. It's not resident, it won't bioaccumulate in the environment. I guess the concern that we have is with birds, that some of the ones we've been finding, there's been bait-like material, so mixed seed, cracked corn, millet, that kind of thing, and potentially mixed with some of this chemical. So it's really when the chemical is not used according to the directions or according to national approval that we have a bit of a concern. So we're just working with the EPA's laboratory to try to develop a test to be even able to measure this in animal tissues and in some of that bait substance. We just don't even have that at the moment, but it's coming on board very quickly. The issue with this is that the chemical, when it mixes with gastric acid or even moisture, it starts to undergo a chemical reaction, and it creates zinc phosphine. So it goes from phosphide, a solid, to phosphine, which is a gas. And it's that gas that is a potent rodenticide, but if others are exposed to it, it's really nasty as well. So just be careful if you're handling mass mortalities of birds in that area. We'll talk next week about who to call and who to notify in those kinds of events, but you're better to make that phone call rather than handle these animals yourself. We care for you and want you to care for yourself as well. So intoxication events, these are how the birds approach. They present in a very typical way where we have groups of birds, sometimes multi-species, often multi-species, certainly more than normal, not something you'd expect to be a usual event. They're in excellent body condition. They're well hydrated, and look at this, they've got a crop full of seed, and it's not the kind of seed a galah would normally eat out in the wild. It's usually something that looks like what you get at Coles in the pet shop, the pet area. So when we collect those kind of specimens, we put them into the glass jars on the right, which are specially prepared hexane washed glass jars that the forensics lab uses for their analysis. Or sometimes if we don't have any jars left, we'll wrap the specimens in alfoil and then put them in plastic. But we have to make sure we're not putting them in plastic tubes or vials where some of the chemicals in the vials could contaminate the sample. So it's very particular processes, I suppose. And down on the bottom, these are all the cryo vials. So we take a bit of liver, lung, kidney, spleen, brain, and large intestine into cryo vials and chuck them in our -80. So if it's not an intoxication event, we can have all the samples we need to test for everything else. Now, Babesia, you guys have probably come across Babesia before. Am I not screen sharing anymore?
No. Sorry, Kerry, I was just going to mention that. I'm not sure what happened.
That's all right. I'll put it back on. Just let me know if it...
Yes, Babesia. Good to go.
Yeah. Cool. Alrighty, so Babesia, we've been seeing this. Initially, it was something that was started up in the New South Wales, Queensland border up around 1995 period of time. And more recently, it's moved down to New South Wales and south of New South Wales, up into Queensland, mostly eastern grey kangaroos, rare agile wallabies as well. You can see how emaciated this animal is. Usually, in the past, it's all been hand-reared young that might have been a bit immunocompromised because they didn't get much colostrum from their mum, and individual animals, ill thrift, anemia, weight loss. But more and more, when we're seeing artificial concentration of wildlife, we're seeing Babesia. So I'm hoping you can see all the little blue dots that are a bit bigger on the left-hand side. So that's a red blood cell that's exploded as these parasites replicate inside and burst forth. So just a sad situation. And I guess what we're seeing in some areas is in Morisset, we had a massive outbreak with, we know at least 450 eastern grey kangaroos died over a few-week period, but it was likely to be a lot more. And in the lead up to that, with the week before the outbreak, the local newspaper had put out this photo saying, don't feed wildlife, because it was a notorious spot on websites for backpackers to stop up in Morisset and feed the kangaroos. And yes, he is feeding that kangaroo with his mouth. And it is also probably just a natural area because of all the grassland in an otherwise wooded area where kangaroos might like to live. But concentrating like that, we get this parasite. It's transmitted by ticks. You get a lot of hosts in one environment and they're not socially distancing. Get your kangaroos socially distanced. Otherwise, we can see this kind of an event happening. So on the left, this is something you and your vet can diagnose in the local vet clinic. On the left, you can see a kidney impression smear. Kidney is a place where these things like to live, and the little capillaries is a kidney. And then on the right, that's just a squash of some brain, and you can see the capillaries and the little parasites in the capillaries. So these are just diff-quicked off, like in the clinic on the day of the examination. So something you can definitely make a diagnosis locally. And this is something that we've just been starting to see more recently, which is a fungal infection, and it's called Menisiopsis barbatae. It's not a new fungus. It's been reported around the world in captive reptiles as a really nasty, invasive fungal infection. But just over the last few years, it's been recognized in Australia, across Australia, and causing horrific lesions in free-ranging wildlife in 5 different species of lizards. You can see some of the colonies on the right, so we can grow it in culture. It's a really tricky one. You need people who are really clever, like Paul and Natalie, because it's really quickly overgrown by other fungi that just might be on the surface. So I'm hoping you see how horrific these lesions are. Missing lips, missing eyelids, can't see the ear. So a real welfare concern for these animals. And treatment, unfortunately, is really challenging to the point where there's really not good treatment options. So unfortunately, this is what we have to do to get funding to look into some of this stuff, is we have to give it a really sexy name and make it sound interesting. So we called it zombie fungus killing wild dragons, and it was largely, this is a bearded dragon, but it was largely eastern water dragons, through a project that was conducted through the University of the Sunshine Coast, Queensland RSPCA, Biosecurity Queensland, Murdoch University, and ourselves. So I'm hoping you can appreciate that it can be something from really mild, like this little lesion on the chin of this animal, over to the animal on the right, where it's so proliferative and thickened and the animal's emaciated. And I don't know if it lost its foot because of the fungal infection. I really can't tell you for sure. But it's just debilitating for the affected animals, and they pretty much uniformly die. So it's a really sad outcome. It's really important to differentiate these from more common fungi, which are treatable and are less invasive. So culture and getting a definitive diagnosis are really important because they can help you differentiate this from other things. And this is Nicola Peterson, the honours student, who did an incredible job pulling together all of the cases across Australia. She was the one who initially triggered on that some ecologists working up in parks in Brisbane might be seeing the Nisiopsis. So pretty brilliant. She identified a new way to culture them and did a bit of a risk analysis, as well as just identifying the agent down to a really low level. As mentioned before, we work on Christmas Island, so there's some reptiles on Christmas Island. This top one is an invasive reptile. I'm hoping you can appreciate that's not normal. He's emaciated and he's got all these bumps all over his body. It's just terrific. And then the Kardashian lips on the left. And that's from a novel bacterium you can kind of see in the chains over here. So we're very fortunate to have another PhD student, Jessica Aegis, who helped us to identify what this was, a new bacterium. She did some tests to try to develop treatment and determine whether she could recreate disease so that we could determine whether the treatments were working. She sequenced the whole genome, which isn't easy when you can't grow the bacterium. It will not grow in any kind of culture. So getting enough material to do the sequence on, I mean, that was absolutely brilliant. So Jess is now working with New South Wales Health. She's doing some of the sequencing on the COVID, you know, when someone gets affected and they're trying to figure out where it came from, and she's working in that lab, which is awesome. So we try to make sure that all of this information gets out there. So keep an eye out for this bacterial infection if you can, if you're seeing emaciated, lumpy reptiles. It's something we haven't seen on the mainland and we hope never to see on the mainland. But if we do, it just needs to be detected really, really quickly. So please keep out your eyes and ears. And now over to something with less scales and more fur. So we've been seeing, in addition to cases and outbreaks of babesia, so instead of just individual cases, we've been seeing more outbreaks. And sometimes the history is that they're in a national park and they're being fed, and sometimes it's because they're potentially in bushfire areas where the habitat all around them is quite poor. So we've been trying to differentiate those babesia cases from another syndrome that we've been seeing in macropods. So eastern grey kangaroos, swamp wallabies, and redneck wallabies. So it's been intermittent from about June 2019 until now, and certainly last year between February and June, we saw just a remarkable number of animals. So it's males and females, adults and subadults. Often they're found dead, sometimes they're found hunched over, neurological. I can show you this animal. This poor animal, the carers had done a brilliant job of rehabilitating it after bushfires. It had third-degree burns on both of its feet, only to succumb to this. And so it was just about ready to be released after intensive care for weeks and weeks and weeks. And then this happened. And not 6 hours later, the carer went home and another animal had been affected. It doesn't seem to spread through groups of animals. It's just episodic. One animal here, another animal there. And they will often either present like this, really moribund, weak, depressed, having problems breathing, really quite sad to see. Or otherwise they'll present with edema, just like little gelatinous accumulations of fluid, sometimes around the eye, sometimes around the groin, down here in the groin, and sometimes we also see these little hemorrhages. So what this is telling us is the blood vessels are letting go. There's something wrong with the blood vessels. They're not holding fluid and they're not holding cells anymore. This one's a bit more graphic. I hope that's all right. Look away if you're a bit squeamish on this one. But it's just to show you that there's a lot of fluid around the heart. And then in the lungs, we shouldn't see these marks of the ribs on the lungs, and they should be pink and fluffy and frothy. They shouldn't be dark red and progressively more and more full of blood. So again, just another indication that something's attacking those small blood vessels. And it's a process that we have never seen in wildlife before. It's a bit of an autoimmune kind of process where there's an antibody antigen complexes we suspect in those blood vessels causing this, and to try to figure it out is tricky. So we have gone through that metatranscriptomic process, and we've identified 4 things in there, and probably most of them aren't a problem. It's just now trying to figure out which one likely is the culprit here. If any, it could be something completely different. Another macropod disease that we've been seeing more and more in the Sydney Basin. So initially we saw this down, again, 2017 was a busy year. In swamp wallabies, there was a really astute ranger down at Mimosa Rocks National Park, beautiful national park, highly recommend going there. You will see whales very close to shore if you camp there. It's gorgeous. But they came across this one swamp wallaby with total ear erosion and these little cloudy eyes. She also had lumpy jaw. I can hope you can see how lumpy that jaw is, and she was emaciated. So we went down there, worked with the rangers, and we did euthanize her because her outlook was really quite poor, to try to figure out what was going on. And we identified in the lesions these single-celled parasites with little tails called trypanosomes. And we're just trying to figure out whether that just happens to be in the lesions or if it's actually causing the lesions. So here's a more well-developed and a little bit of a close-up, because I've had even veterinarians that I work with really closely say, oh, no, that's not affected. It's just got some tears in its ear. And you can kind of see it's not just a torn ear. There's these really nodular proliferations along the edge and really thickened and undulating ears. It can be a bit more mild. And then the eyes, we get just a little bit of swelling and redness around the conjunctiva. I'm hoping on the top one is less severe, and the bottom one is very, very severe. It's the worst one we've seen, where I think that cornea is actually ruptured. There's a huge discharge. Sometimes we can find heaps of those parasites in that eye discharge, just again, taking a swab and rolling it out on a slide and staining it with Diff-Quick. But there's also some ticks around there. They're little larval Ixodes holocyclus. And just last week, after looking at probably about 30 swamp wallabies, Hannah found little protozoa in a blood vessel in one of the eyes. So we've got a student project planned to try to help us pinpoint those organisms in the lesions to better link the parasite with the lesions. But we're seeing more and more around places like Coasters Retreat, the Basin, other parts of Sydney. So it'll be another student project to try to figure out what the prevalence is and what the impact is on the population. We've also been seeing bats this season. I'm not sure, probably a combination of habitat change after the fires and then really, really wet conditions. We're not exactly sure what's happening in the bats, to be honest, but we've had a lot of young bats and subadult bats presenting, and they seem to go into care, and really expert carers like Meg Churches and Sarah Curran and others who do a brilliant job. They've just been finding they don't go downhill really quickly. They come into care, they're in excellent body condition, they've got fat and muscle and well hydrated, and they die within 6 to 12 to 24 hours max. What we've been finding is, I'm hoping on the right side, you can see this is an impression smear of the lung. See how much we do without having to do histopathology? This stuff is actually really quite cheap, but it tells you an awful lot. We're finding bacteria. So we've been finding the same bacterium in a lot of those flying foxes. And it's a kind of an opportunistic bacterium. It's not the kind of primary pathogen that chlamydia or tularemia would be. So we're not sure if something else is happening in the bats that might make them a bit more predisposed to this pretty common dirt-borne organism. Just something we're working on right now to try to figure it out. Another puzzle. We've been really fortunate to work with the rangers at North Head for a long period of time on the penguins and the bandicoots and a few other areas across New South Wales as well. And sadly, 3 of our long-term wildlife health study sites have been hit by bushfire. So we were fortunate that we had pre-bushfire samples to run that metatranscriptomic process on. We've now got samples from each of them, but we only have the results from one, which is down at Naji National Park. And we've identified 2 novel viruses that we didn't identify before the bushfires. So I guess this is sort of our first indication, I think, in real time. There's a big theory that anthropogenic change causes the emergence of microbes that we wouldn't have otherwise seen. And I think we're just picking it up in real time for the first time here. We've also been worried about human health and the potential tick-borne diseases. So we've run those kinds of samples, ticks, all sorts of ectoparasites through the metatranscriptomic process, and come up with about 18 different RNA viruses. And one of them is related to cultivirus. It's a cultivirus related to Colorado tick fever virus, which is a tick-borne disease in North America. And it's also closely related to EH, which is a European tick-borne disease. So that plus the tularemia, there's probably other things out there. We don't have Lyme's disease, but there are other things, and we just need to better understand what the ecology of humans, animals, and some of these microbes. So just some useful resources, which are definitely the codes of practice for injured, sick, and orphan native animals, all sorts of them. So there's been a tremendous amount of work put into preparing these and pulling them together. And if you just Google those terms, you'll find them. Or if you look on the recording, you can follow the web. The Australian Registry of Wildlife for Health, we have a web page. We're just updating a lot of the resources. So Jane has done a brilliant job redoing the common diseases of urban wildlife, and that will be posted in the next few weeks, along with videos from our last wildlife pathology short course, which pretty much summarizes all of the diseases of all of the animals. And then the website from Wildlife Health Australia is just resource rich as well. All sorts of fact sheets on individual diseases, wildlife health report, national guidelines for management of disease in free-ranging Australian native wildlife, so Australian wildlife, just a wealth of resources there. So please check that out. And I finished. So hopefully there's a few questions out there. Sorry, I took a bit longer than I hoped.
No worries at all. Thanks, Kerry.